So chapter 22, we built up fats, and we've already built up sugar, carbohydrates in the sense for gluconeogenesis. Now we're going to do the biosynthesis of amino acids in particular. We'll talk a little bit about some of the other related molecules, but we're going to focus primarily on amino acids for time's sake. So that way, hopefully we can finish the semester out by looking at the neurotransmitters and hormones and everything, which is the last chapter that we'll be covering. This semester, that seems like it's never ending. Word, that kind of stuff. So yeah. All right. So just like breaking down the amino acids, we focus on the nitrogen because you already know how to get rid of the carbon and oxygen. Ultimately, it's going to be carbon dioxide. Right. But a lot of this has to work with how we use urea cycle to get rid of the nitrogen, but how do we get the nitrogen in? Okay. So from where do we originate this nitrogen? And it should make sense. Behind carbon, hydrogen, oxygen, only nitrogen is the next greatest in contribution to mass. Usually I have my computer up to where I can see ahead, since the Mac won't let me look to see what's coming up. So I apologize if I repeat things. Oh, whoops, I went too far. More than normal. Many times amino acids and nucleotide biosynthesis are linked together because we can make amino acids and then utilize part of those amino acids to make the nucleotides. Okay? So that's why they are very related. Typically, you're going to have the transfer of nitrogen. Nitrogen is really important, and then the transfer of carbons, especially carbons at 1.9. And there are so many genetic diseases associated with these pathways. So many that we can. Some of you are even writing papers about this kind of stuff. So we'll only touch upon a fraction of those. Oh my goodness, my ears are like popping like crazy. Alrighty. This is the nitrogen cycle. Okay. And I want you to look over it. Normally, comments that pop up. That's why it would be a big help if I could get this computer to work. I don't remember the order with which I cover it. Now, so now I know what part we're going to start off with. So how do we get the nitrogen into where we need it from? Because obviously we're wanting it to make it in terms of amino acids. So where does it come from? First of all, I want to point out that nitrogen, and I've actually asked this question before, not meaning it to be difficult, like, what's the primary component of our air? And people say oxygen, but in reality, it's not oxygen. Actually, 80% of the air is nitrogen too. Okay, so there are bacteria and microbes that will fix it in order to take it from the atmosphere and incorporate it in the form of ammonia, which can be used. Okay. And so that's why we have these nitrogen-fixing bacteria. We also have archaea that can do this as well. And it utilizes the nitrogen from the air and then to make ammonia. And then plants can, this got too big. So then plants can utilize the ammonia and incorporate it directly into amino acids. Okay, and then so therefore we can either get it from our plant-based diet or from eating animals that eat plants. That then make it to make proteins, and we can get it that way as well. So don't go and say that you have some like a hippie-loving vegan professor here saying that you can only get your nitrogen from plants. You can't get it from animals, but those animals got it from the plants. They got it from the bacteria. And so, not that there's anything wrong necessarily with veganism, so we'll go the other way. All right, so on the other end of the spectrum, and one thing I wanted to point out here is if you go back to Gen Chem, the oxidation state for N2 is 0, and so you can either have it where it becomes reduced versus, and this is its fully reduced state, right? Because you've totally increased it in the form of nitrogen and hydrogen bonds, and the fully oxidized state is nitrate. Okay, so it's similar to carbon. You increase the number to oxygen bonds, and it's going to be oxidation. You increase the number to bonds to hydrogen, then you're reducing it. Okay. On the other end of the spectrum, we have denitrifying bacteria. in other organisms. And what they can use is they can actually take nitrate and utilize it to where they can then reduce it down to nitrogen, okay? And so essentially what they do is instead of using oxygen to make ATP, you know how we use oxygen to make water? They use nitrate to make nitrogen, and that's how they, as part of their electron transport chain, an oxidative phosphorylation, okay? And so, and we utilize that. It's great for the ecosystem, so that way that we can have, it's very symbiotic in the sense that we can make nitrogen. Not we. They make nitrogen for us ultimately, since other things can take that nitrogen, turn it to ammonia, and then we can utilize that to get our amino acids. Okay, now this is a little dated because I made this slide a few years ago. So each year that passes, it's not quite as astounding, but I said because the term recently is relatively speaking. Okay, it's not like in the last year, but it's in the last, I don't know, number of years, few years. At least within my lifetime. So it's really recent then. It's okay to laugh. All right, so what happens is there's an actual class of bacteria that they now think actually converts the bulk of nitrogen in the biosphere, okay, to either NH3 or to nitrogen, to atmospheric nitrogen N2, okay? And so that's called the Anammox bacteria, which is an awesome name. And just like we have, we have a compartment that will, utilize oxygen, and we get energy and everything from that. That's our mitochondria. They have a compartment. It's not mitochondria because it's a bacterium, right? So therefore, it doesn't have mitochondria. But they actually have a compartment, a lipid-based compartment that does this. The Anammox bacteria does. Do you know what that compartment is called? You know how biologists like that zome, like chromosome and yeah. So this one's called the Anammoxosome, which is a mouthful. Okay. But I mean, it should look a lot like. It kind of should give you that idea of the electron transport chain, the oxidative phosphorylation that we see in the mitochondria. We have this ATP synthase going on. It's very similar kind of thing. It's just that it's not utilizing oxygen. It's utilizing nitrogen. Okay. And so what happens here, this would be similar to the matrix, mitochondrial matrix. And of course, this is their cytosol. They can take ammonia, and they transform the ammonia into hydrazine. Hydrazine is rocket fuel. Okay, so yes, it is very explosive that this can happen. If you notice, this is completely anaerobic. So that's why they don't have to have O2 at all. You don't see O2 there. Okay, the oxygen that they get is actually from nitrate or nitrite. All right, and this hydrazine oxidizing enzyme is what converts it from hydrazine to NH2, and in the process, you get a whole bunch of protons out of it. The protons, just like what we saw before in our mitochondria, utilize it, go through the ATP synthase in order to turn ADP into ATP. Okay. Then on the other side, they take the protons from one side along with the NO2 to make water and ammonium hydroxide. Okay. And that's what gets converted, once again, to make hydrazine, and it goes on and on and on. This is anaerobic. It was found originally in sewage. Okay, and so, because in all the crap, literally, that there's no oxygen getting down to it. So this is one way that they actually utilize nitrogen, and in the process, convert it to N2, which would be a gas, which then can be released into the atmosphere. So, see, this kind of crap is important. I mean, yes. I'll be here all week long, folks. Okay. Maybe they need me to be the opening act for the keynote speaker on Friday. Mm-hmm. Yes?
What are those four electrons used? Are those used to are they used by the enzyme?
Yeah, because remember, reducing means it's gaining electrons.
Oh, yeah.
And so that's where the electrons come from. Just like we have an electron transport chain, that's essentially their version of electron transport chain. A little simplified. And it's relatively recent when it's discovered. But, I mean, you don't want to let a match run around it. Boom. Okay, so. All right. So yeah, the conversion of nitrogen to ammonia is very, very exergonic, meaning you have lots of energy. But N2 is very stable. Okay, that's why you don't have to worry. You know, you can have your candle in the wind. You know, play some Elton John up there, without having to worry about the entire atmosphere exploding. It's not faith integration, but it's pop culture integration. All right. Now there are bacteria in the archaea that's going to actually utilize this. And in the part that has the boxes, this would have been a box. So just a little FYI. That the complex that they use to transform all this, so that way we can get it, make it, it's called nitrogenase complex. So it makes sense. There's ACE and enzyme, it makes nitrogen, so it's called nitrogenase. It's huge.
They use the same TPP like we do with the complexes.
No, this one's different. I'm going to show you in just a moment. It does utilize something similar to some of the complexes that we have, but it's not TPP. Okay. All right. So knowing this has to do with the redox reactions, right? So you're moving electrons around. What were some of the ways that we moved electrons around? Within our complex, 1, 2, 3, 4, that kind of stuff. What?
Iron sulfurs.
It's iron sulfurs, okay? Yep. And so that's why, and yes. Now there is a couple of unique facets to it, but there are some things that look very, very familiar. Okay. So here, if you notice, they take something that's relatively common to all organisms, pyruvate and coenzyme A, and they're ultimately turning it into acetyl-CoA, which we've already talked about how the acetyl-CoA for these, well, even for us, but also for these organisms, can be used to make energy, right? So, it can enter in for the citric acid cycle, so on and so forth, or be used for, installation of various things. Okay. But in the process, it gives off a bunch of electrons. So that's how to read this. So you can, it doesn't matter to me which that we start with, but we're going to start down here. So we take pyruvate, it's going to start returning the pyruvate into acetyl-CoA. Of course, we utilize, what enzyme complex do we have that turns pyruvate into acetyl-CoA? Good to know for the final.
EDC?
Yes, pyruvate dehydrogenase complex. Okay. This one, the first recipient of these electrons goes, and once again, if you notice, it's always going to go from oxidized to reduced. Since it's gaining electrons, it's being reduced. It's ferredoxin, and it's fine. Or, depending on the organism, some of them are flavodoxins. It means it has a flaving group and stuff. Ferredoxin, okay. Now, either the ferredoxin or flavodoxin, depending upon the organism, it's going to have to be regenerated. So it's going to give off the electrons to the next partner in the chain, which is this group of 4. Please notice the next slide will have all 4 of these shown. Okay. Here we go. Denitrogenase reductase is the next enzyme of the chain. And so sorry about that. It was oxidized. It gets the electrons from the ferredoxin to become reduced. It takes a whole bunch of ATPs. I would never ask you to memorize the fact that, if there's 8 electrons, you have to use 16 ATPs. It takes a whole bunch of ATPs, and then it's going to cycle through. You have the the cleavage, the hydrolysis of the ATP down to ADP, ultimately, for this cycle. It's passing it off, and the partner that it passes it off to, and that's the dinitrogenase reductase. Whoops, it went all the way through. I'm sorry. Here, let me go back. Is dinitrogenase. So it should make sense that dinitrogenase reductase is reducing dinitrogenase. See, because since it's reducing dinitrogenase, this is dinitrogenase, and it goes from oxidized form to reduced form. It's going to pass off the electrons, and it passes off the electrons from nitrogen to make ammonium. Okay. And then plants can take the ammonium and incorporate it into the what should we call it? Amino acids. So we have the ferredoxin or flavodoxin. That goes to the next one, which is dinitrogenase reductase, which does utilize ATP. And then dinitrogenase reductase reduces dinitrogenase. Dinitrogenase takes the diatomic nitrogen into and turns it into ammonia or ammonium. Then we're going to focus a little bit on dinitrogenase. It's large. And one of the reasons why it does utilize some of the things that we've seen before, like iron and sulfur, but it has something very, very different to it than what we've seen. What's MO? And it's not Missouri, my home state. But it is, but not here. So, what is MO? What? Molybdenum. Okay, it's another metal. And so it's just one that it's important in vivo, but we don't get to see it very often. It uses molybdenum. Okay, and so this dinitrogenase is huge. It has iron and molybdenum at each active site. It's a tetramer, but it's a dimer of dimers. That's what that means, 2 plus 2. So we have a dimer and a dimer together makes up a tetramer. And per tetramer, you have 2 molybdenum, 32 iron, 30 sulfurs, and so it's huge. And there are a handful, at least, of organisms that instead of molybdenum use V, and V would be vanadium. Okay, so that's why, just a little side note. So this is what it looks like. Let me make this smaller. I have problems seeing this, but hopefully you guys can tell the difference in the colors. So I got it from your book, and so I wrote it down, so I'm assuming that they are true. But dinitrogenase are gray and pink. I know it's supposed to be the inside. And then the reductase, which is huge, you know, it makes a big complex formation, are supposed to be on the outside, which I can see the blue and the green on the outside. Okay. Then they're showing where the ADP is bound, because if it's ATP, then the reaction would occur. But so this is ADP, and then it shows that iron and sulfur and the molybdenum. So you can see this, they're really big. So if we take away just the proteins, you know, it's kind of hard to tell where everything's at, but if you peel away the, boom, there you go. Now we can see how iron and molybdenum and everything is related. And so you can actually kind of see the way that the electrons are going to be flowing. Whoops, that's not the right one. That we're going to be able to flow the electrons. Like, so before, if we look at it, proteins, then we can see how ADP, or it should be the binding site where ATP would also be, is with respect to the transfer chain for the electrons. Then if we just blow this part up, then we can see how iron and sulfur all hold that molybdenum in a certain configuration in order for it to occur, which once again, that's called chelation therapy. Coordination numbers, and so, or chelation science, the way it's held in place. Okay. I would just want you to know that it uses molybdenum, or that some organisms, instead of the black, is be vanadium, which is a different, and iron sulfur, which is what we've seen in the past. Okay, so the next questions that we're going to start to address is how the ammonia gets incorporated into the actual biomolecules. We got into ammonia now, from nitrogen to ammonia. And we actually got it from nitrite or nitrates in a moxazome to make it into the N2, then goes into the ammonia. So now we got to get from the ammonia into the biomolecules. Okay. So then we have to think about what possible amino acids can be used for this. You can't just say all amino acids, because I mean, yes, it's true, all amino acids have nitrogens. But what do you think, knowing what you know about how the breakdown occurs and to shuttle, to move nitrogens around, what amino acids do you think make the most sense as ports of entry for nitrogen in the, you know, in the plant system? What? Glutamine? Yes, glutamine is, and there's only like 2 other possibilities that we've talked a lot about, like No, not lysine. What's another one that's used a lot of times with different types of shuttling for nitrogens? Not asparagine, it's glutamine and glutamate. Okay, so glutamate. The third one that kind of wouldn't make sense, it's not true here, is alanine. Remember, our muscles utilize alanine, but plants aren't not like flexing muscles, so we don't have to worry about that. But yeah, so it's glutamine and glutamate. Okay. So, and that's true, it's most organisms will take this, they contain specific enzymes that will take that ammonium to make glutamine. What enzyme do you suppose, I think I put the block on it. Let me just check. Yes, I did, good. What enzyme do you suppose is doing this? So if we look at it, it's a two-step process. We have glutamate. We have ATP. You first activate it by putting the phosphate on the very end of the glutamate, because that's where, remember, the only difference between glutamate and glutamine is one's a carboxylic acid on the end, and glutamine is an amide. Okay, so we put a phosphate on it first, because the phosphate's a great leading group, and then we can go ahead and take that nitrogen that we're wanting to add into our system and have it pop off the inorganic phosphate and get glutamine. So what class or classes of enzymes could this be? Well, I can see transferase, possibly. Okay. A what? Maybe a kinase. What's another class? This what? Oh, synthetase. So which one of all of those possibilities do you think makes the most sense overall? It's a synthetase. This is just showing you the intermediate that's formed. It's a ping-pong reaction. If you think back last semester. This is synthetase. What specific synthetase is it? It's glutamine synthetase. That's why it's not. That should make perfectly logical sense. It requires ATP for energy. We say for energy. That just means you're adding the phosphate. Usually it's the phosphate. Sometimes it's the A part, but usually you're adding the phosphate to something. We're just adding the phosphate to the glutamate in order to have a better leaving group. And so what I mean by that is it's going to look like this. Don't get confused by that gamma. Does anyone know what does that gamma mean? See, it says gamma glutamyl phosphate. It's not a confirmation, and that's a good guess. What? It's not meaning that it's an intermediate. It's what carbon it's on. It's the position. Remember, we have alpha and beta, and so if you go far enough down the Greek alphabet, that's what that means. It just looks kind of weird because we're not used to seeing gamma as often as we are with alpha and beta. So 1, 2. So, this was normally what glutamate would look like. That's supposed to be negatively charged. Okay, and so that would be the gamma glutamyl phosphate. It's just got that phosphate there. So now we can see where nitrogen, which would be a nucleophile, would come, smack that carbon right there, and pop off the inorganic phosphate. And so that's how we make glutamine from glutamate. Okay, that being said, plants and some bacteria can also do something else that we don't do. This is just really more for a point of notification. This is what we would talk about with respect for us if we're just trying to incorporate using nitrogen. They can actually use glutamate synthase. That's why you have to be very, very, very specific, which we cannot do. Oh, that's supposed to be alpha. The alpha got cut off. But that's where you take alpha ketoglutarate, you take the glutamine, it takes NADPH. Oh, it's got all cut off. Let me see if I can get it. And you get 2 glutamates out of the process. Okay. And so it's just a way that, that you can also transfer the nitrogen along without using an actual amino transferase. It's just like a little side note there. I'll just give you a hint. Since we can't do it, and and typically this isn't medically relevant, there's a 99.9999% chance I will not be asking about it. So, okay. All right. So let's get back to glutamine synthetase. It's regulated. It's hugely important. You can imagine, since nitrogen. We talked about the importance of getting rid of nitrogen when we no longer need it, because otherwise you just had ammonia build up, you would die. It affects your pH, it affects osmosis, osmotic pressure, and everything. The glutamine synthetase, since it's involved in nitrogen regulation, it's tightly regulated in all organisms. It can be covalently modified, and it has 8 different allosteric inhibitors. Okay, and so none of them completely turn it off. So this is a great example how, like the light switch either being on or off, or that kind of a dial. Like if you have all 8 on there, it's off. If you have 6 of the 8 on, it's 3/4 of the way off. That kind of thing. So it's a nice way to be very precise in the level of nitrogen incorporation. Okay, so they are additive. So this is what I mean by that. One, two, three, 4, 5, 6, 7. Yeah, we got all 8 on here. Okay. And it shouldn't make sense. So remember, we're going from glutamate to glutamine. It takes in ammonia, and it does require ATP. That's why it's a synthetase. But glycine and alanine inhibit it, which should make sense in the sense that if you have a lot of amino acids around, you don't need to be adding more nitrogen in, right? Other things that inhibit it are also histidine and tryptophan, 2 more amino acids. CTP, what does it make sense about CTP? What is CTP? Or AMP. I mean, you can utilize both of these. What are CTP and AMP? What? That AMP is adenosine monophosphate. What about CTP? It's not cysteine. Cytosine, right? But what class of biomolecules are they?
Nucleotides.
They're nucleotides. So just like if you have amino acids, a lot of amino acids, you don't need more nitrogen. If you have a lot of nucleotides around too, right? That's where nitrogenous, those are called nitrogenous bases. Like all of our bases that we use for nucleotides are nitrogenous bases. So if you have a lot of them around, we don't need to be having more nitrogen coming into our system. And then actually let's go to glucosamine. Glucosamine 6-phosphate is also an inhibitor. What class of compound is that?
It should be plus A.
Mm-hmm. The sugars are big biomolecules are carbohydrates. So once again, and this is a nitrogenous carbohydrate, like glucosamine. So therefore, if we have lots of it, we don't need to be adding more nitrogen. And then lastly, carbamoyl phosphate. We have seen this in one place. Where have we seen carbamoyl phosphate? It's towards the beginning of the urea cycle. If you have a lot of it, that means you've got more than enough nitrogen. Your body's trying to get rid of it, so don't be adding more nitrogen into your body in the form of glutamine. Okay, so that's why it kind of makes sense if you break it down. Yes, these are very specific amino acids or nucleotides, but if you just think about logically, if you have too many amino acids, too many nucleotides, too much of the nitrogenous carbohydrate, or the urea cycle's in overdrive, you don't want to be making more. That would be a futile cycle overall. Okay. Are there any questions overall on any of this? Okay. Because we're definitely going to stop here because the next Other processes involved here, we have alpha ketoglutarate going to make glutamate. Of course, that's using an aminotransferase. If you have lots of alpha ketoglutarate, then you're going to actually activate this process here, and glutamate can go on to make glutamine. Of course, we've seen that already, and that's utilizing glutamine synthetase. And once again, if we have too much glutamine, it comes through here and it's going to inhibit this. Inorganic phosphate also inhibits the uridylation portion as well, because it is one of the signifiers that we have plenty of it around. Okay, so it may take a little while for you to look through this and to try to figure out what's going on, but I do suggest that you do just remember that P2 is regulatory protein, and what makes it so different than what we've seen in the past is it both activates and inhibits the other enzyme, and it's based off of whether or not it has been uridylated. Okay. All right. So we've already talked about some of the different classes of reactions that occur in biosynthesis, like aminotransferases. And we've utilized PLP for racemization and decarboxylation as well. There's also now another class that can occur, which is called a 1-carbon transfer, and it uses either tetrahydrofolate or S-adenosylmethionine. S-adenosylmethionine is one of my favorites because of the fact that it's got 2 nicknames. Does anyone know what their nicknames are for S-adenosylmethionine? Sam and Adomet, which to me sounds like something from Star Wars. And so for adenosylated methionine. Okay. So yeah, Sam or Adomet. And the aminotransfer from amide, for the amide nitrogen glutamine, there's lots and lots of different reactions that actually utilize glutamine as its major source. Okay, so that's almost like, if you had to guess, stone. But if you had to guess, with respect to nitrogen, glutamine and glutamate are the 2 biggies. Alanine with respect to, muscles getting rid of it to a different, ultimately to the liver. Right? So one class in particularly important is called glutamine aminotransferases. Okay. So this enzyme is going to be transferring the amino groups, but yes, it's off of the amide nitrogen, okay? There are 2 domains, so it's really cool. It's actually quite remarkable in that one of them is going to bind the glutamine, and the other one's going to bind whatever, the other domain binds whatever substrate is going to ultimately get that nitrogen. And it's really nice because that nitrogen never leaves the enzyme, so you don't have to worry about ammonia. Since remember, ammonia is very toxic. We've already talked about the fact that it messes up the osmotic pressure and the pH as well for various tissues. Okay. And it's believed that cysteine is the nucleophile that does this. But the nitrogen gets channeled through, and they can call it the ammonia channel. This is what it looks like. So I guess technically I want you to know this mechanism, but it's actually a very, very simple mechanism. Okay. Because cysteine is a thiol. Whether it's a thiol or thiolate, it's a great nucleophile. We have glutamine here. From organic chemistry, you smack that carbon. It's easy. It's a great electrophile. Technically, you can even form the tetrahedral intermediate, which isn't shown here, but you get collapse of it, and it's the nitrogen it leaves. It does pick up a general acid in the process, so it leaves as an ammonia. Okay. This ammonia then is going to go on through that ammonia channel and to an awaiting substrate. And it's either going to be some type of alcohol or some type of ketone or aldehyde. Okay. I said alcohol, but technically it's a phosphate. So depending upon what you have here, if this was like a phosphate, then of course the nitrogen goes, attacks the R, and it pops that off. So you would get inorganic phosphate as a product, and you have an amine now. If it's an aldehyde or a ketone, you go through and you're going to make an imine. Okay. And so, and you get water off. We've seen that kind of mechanism over and over again. It smacks it here. Technically, it would smack it twice since you have a double bond there, and it would pop off water, and you have an imine. That's why I say it's very, very simple mechanism. And so that's it. Are there any questions over the glutamine amido transferase is just another way that we can actually move and shuttle that nitrogen from to lots of different things, lots and lots of different things. Yep.
So is this a suicide enzyme, or does it ever come out?
Oh no, it does. It does. They showed it here. I guess one bad thing about biochemistry, especially later on in biochemistry, is they just assume that when you see this water, that water has it shows water coming into that glutamate, they're just assuming that you realize that it kicks it off of the enzyme. Because what happens is this water, here I'll draw it in. The water, of course, is a good nucleophile as well. It comes and it smacks the carbon and pops off the cysteine. And so then you get glutamate is always going to be one of the products because you've deaminated. Sounds great faith integration of some sorts. So I've been to a few of those kind of sermons. So, okay. Are there any other questions? Like this is the very end for part 1. I want to get started on part 2.